Today we're going to talk about phospholipase C and protein kinase C signaling. If you look over to the top left corner, you're going to see our G protein coupled receptor. G protein coupled receptors, or GPCRs, contain 7 transmembrane alpha helices. They translate the binding of extracellular signaling molecules into the activation of GTP binding proteins. GTP binding proteins, or G proteins, are connected with vesicle budding and fusion, microtubule dynamics, protein synthesis, nucleocytoplasmic transport, and transmitting messages along cellular information circuits, which we are going to cover in this video. They constitute the single largest superfamily of proteins encoded by animal genomes. And examples of natural ligands that bind are a diverse array of hormones, neurotransmitters, opium derivatives, chemoattractants, odorants, tastants for smell and taste, and photons. In the case of the effector phospholipase C, which I am covering in this video, example ligands would be IgE antigen complexes, which bind to a mast cell IgE receptor, the physiological response being secretion, or the F-MET peptide, which binds to a chemotactic receptor, the physiological response being chemotaxis. The amino terminus is present on the outside of the cell. The 7 alpha helices traversing the plasma membrane are connected by loops of varying length, and the carboxy terminus is present on the inside of the cell. The inactive conformation of the GPCR is stabilized by non-covalent interactions between specific residues in the transmembrane alpha helices. Ligand binding disturbs these interactions, causing the receptor to assume an active conformation. The active conformation is obtained through rotation of the transmembrane alpha helices, consequently changing the conformation and leading to an increase in the affinity of the receptor for the binding of GTP, thereby forming a G protein complex. Heterotrimeric G proteins consist of alpha, beta, and gamma subunits. I'm going to move on to phospholipase C, which is next. When acetylcholine, which is an organic chemical that functions as a neurotransmitter, binds to a smooth muscle cell, or when an antigen binds to a mast cell, the bound receptor activates the heterotrimeric G protein, which activates the effector phosphatidylinositol specific phospholipase C beta, situated at the inner surface of the membrane and bound there. This catalyzes a reaction that splits the phosphoinositide into 2 molecules, inositol 1,4,5-triphosphate, or IP3, and diacylglycerol, or DAG, both of which are second messengers. Second messengers are small substances that typically activate or inactivate specific proteins. Diacylglycerol, or DAG, is a lipid that remains in the plasma membrane following its formation. From there, it recruits and activates effector proteins that bear a DAG binding C1 domain, the best studied of which is a family of proteins called protein kinase C, or PKC. Protein kinase C phosphorylates serine and threonine residues on a wide variety of target proteins. Protein kinase C isoforms carry important roles in cell growth and differentiation, metabolism, cell death, and immune responses. In studies with a group of powerful plant compounds called phorbol esters, which resemble DAG, these compounds activate protein kinase C, causing them to lose growth control and behave as malignant cells. When the compound is removed from the media, the cells recover their normal growth properties. Cells that have been genetically engineered to constitutively express protein kinase C exhibit a permanent malignant phenotype in cell culture and can cause tumors in susceptible mice. Inositol 1,4,5 triphosphate, or IP3, is a sugar phosphate, a small water-soluble molecule capable of rapid diffusion throughout the interior of the cell and binds to a specific IP3 receptor located at the surface of the smooth endoplasmic reticulum. The smooth ER is the site of calcium storage for many varieties of cells, and the IP3 receptor functions as a tetrameric calcium channel. Binding of IP3 opens the channel, allowing calcium ions to diffuse into the cytoplasm. Calcium ions may then bind to various target molecules, triggering specific responses. Contraction of a smooth muscle cell and exocytosis of histamine-containing secretory granules in a mast cell are both triggered by elevated calcium levels.